Assalamualaikum warahmatullah. My name's Ahmad Mannan. I'm currently a postdoc in the Department of Maths at Imperial College London. As we're just waiting for the computer to become unlocked, just introduce you a little bit into my field. So I've been looking, my background is in maths, but I've been looking at problems in biology, and the problems that have really drawn my attention are those in trying to understand the design principles of regulation in cells. That can happen at the genetic level or at the metabolic level. And in this talk, I'll go through some of those examples, especially those that have inspired me and most recent research into those areas as well. So bacteria, which is the organism that I'm going to be focusing the talk on today, bacteria are one of the simplest forms of life on this planet. But they're composed of a complex network of chemical interactions which make up the cell. The cells take up nutrients and they break these down by a process of chemical reactions called metabolism. And these produce the growth of the cell. They help the cell to grow. They produce energies in the cell and chemical products as well. And these chemical products can help towards creating a microenvironment which allows cells to be in symbiosis with other organisms around it, or even to fend off competition. Now, each of the chemical reactions which happen inside metabolism are catalyzed by enzymes, which are actually proteins. And these proteins are encoded for in the DNA of the organism. Now, cells, these bacteria, and any cells actually, they live in a constantly changing environment. And so they need to be able to sense the environment and adapt accordingly. And there are many regulatory mechanisms that happen on the metabolic level, on the genetic level, on the protein level. And in this talk, what I want to go through is I want to go through some examples in the literature, just a very few of them, just to give you an idea of how people in systems biology and synthetic biology are using mathematical models to try to understand and elucidate what these mechanisms are doing, how they're doing it, and what their purposes are. Now, cells growing in a certain environment, that environment can fluctuate, though it's on short time scales, it can fluctuate quite drastically. The media can change from one composition to another composition, and this can be very detrimental for cell growth unless the cell is able to adapt and then grow in the new environment. Now, one of the first studies actually on this kind of mechanism of how the cell is able to adapt was on the Lac Operon. I'm sure for all the biologists out there, you probably know the system very, very well. But for those who don't, in essence, what it is there's a sugar called lactose, which can become available in the environment, and the cells can uptake that and metabolize it and grow from that. Now, if lactose becomes available in the media, it can be taken up by the cell and converts into allolactose, and that actually binds onto something called Lac I. And Lac I, what it's doing, I'm sorry that the ladies can't see this, but what I'm pointing to is a Lac Operon just above the Lac I. What Lac I is doing is it's regulating the expression of genes Lac Z, Lac Y, and Lac A. And what that basically means is that it's actually inhibiting the expression of those proteins from the genes. But when allolactose is introduced, when lactose becomes available, it actually binds onto Lac I and represses its activity, which means that Lac I can no longer repress the expression of the genes Lac Z, Lac Y, and Lac A. And so what happens is that when that repression is relieved, then the genes are expressed and then they can produce Lac Z and Lac Y. Now Lac Y, what it does, it goes and creates a permease, which is the gray boxes that you see at the top. What that does is actually allows the uptake of lactate. It allows for lactate to be taken up. And if more of those are produced, then there's a greater flux of lactose which is coming into the cell. And LAC-Z, what that functions to do is actually break down the lactose and metabolize it and then use it for the cell. So what's really happening here is that that when there's no lactose in the environment, then the cell doesn't need to produce these enzymes to break them down. Because if it does produce these enzymes, it is actually draining resources from the cell. This I'll tell you the details of later, but in essence, it doesn't need to produce those enzymes. And when lactose does become available, then it does need to produce those, and it needs to metabolize those. And so it needs to produce these according to the demand. That was the work that awarded the Nobel Prize to Jacob, Loeff, and Monod in 1965, where they uncovered, in essence, the first example of a design principle that you're controlling gene expression to produce only what you need and when you need it. And what you need, meaning the proteins that you need to break down and metabolize the sugar, and when you need it. And this opened up a whole field of research into the regulation of genes and the need for expression, and the complex metabolic network that I was showing you before. Not all the reactions are on all the time. Only some of them are on at some times, and others are on at other times. So this was one of the first works that actually inspired my work to look into models. Now, another example of that is not only for the expression of genes for uptake and metabolism of sugars that are directly from the environment, but it can happen inside metabolism. So the figure that you see on the right there, that is central carbon metabolism of the cell. It breaks down glucose and produces and allows the cell to grow. But there are two regulations happening here, two key regulations, the PDHR and the ICLR. And what those are in essence doing is that if there's lots of glucose, then there's lots of this biochemical called pyruvate, and that regulates the activities of these, they're known as transcription factors, which regulate genes. And those are basically allowing metabolic flux to happen like this. But if glucose becomes depleted, those transcription factors, they change gene expression in such a way that the whole metabolic flux distribution changes, which means that the cell adapts to consuming an alternative sugar, which is acetate, and it needs to express the certain genes to be able to take that up and break it down. And it's done through this regulation of genes by the interaction of, by sensing, whether a certain biochemical is there or not, and then changing gene expression accordingly. Now, the enzymes which happen in metabolism, they need to be constructed somehow, right? And when transcription translation happens, you produce a protein, which is the enzyme, and that catalyzes the reaction. But these proteins are constructed from building blocks called amino acids. And amino acids are also produced from cell metabolism. But instead of now talking about the change and the shift that's happening in the environment, there's actually a change and shift of demand of amino acids inside the cell. And the cell needs to regulate the amount of amino acids that it's making to be able to cope with the demand to produce the necessary proteins and the enzymes when there's a shift in metabolism. Now, in this paper on the top right, Chubakov et al., they use mathematical models to try and understand the regulation of amino acid biosynthesis. And amino acid biosynthesis, in essence, what it basically means, the top left figure shows here, that you have an unbranched cascade of chemical reactions, which from some precursor produces amino acids. And then amino acids, if there's lots and lots of them, that actually feeds back and inhibits the first reaction step of the whole cascade. In a way, what it's saying is that if there's lots of amino acid, I don't produce any more because we're meeting demand. But it's a lot more involved than that. It's not just the biochemical product at the end, which is feeding back information, but the figures at the bottom here that you see, there's a transcription factor involved, And this transcription factor, it basically senses the concentration of some intermediate, which is given by the red arrow or line that you see here in 3 and 6. And this, when it senses that by binding to it, it then changes the activity or the expression of the genes for the enzymes that are along that cascade pathway. Now, what Chubikov et al. Did was try to understand what is that doing? What is that regulation really doing in amino acid biosynthesis? And there are many, many amino acid biosynthesis pathways, and they have different, these are called the architectures. These are different architectures of the regulation. Now, one main architecture motif is the one that you see on the left. And that one is, it senses an intermediate metabolite, so an intermediate from the production line. And the one on the right is the one where it senses the product. So the one on the intermediate is sensing some intermediate, and it's saying, okay, I need to change the production flux accordingly. And the one at the end is saying that if I have enough product, basically, down-regulate, because it has an inhibition sign on the right figure, the red inhibition. The red line is an inhibition. Inhibit, the flux through the production. So what Chubikov et al. Did, they actually looked at all different combinations of where the transcription factor can sense from, which step in the pathway the transcription factor is sensing, and try to elucidate, how that changes the dynamics of the flux that goes through that particular pathway. I'm not going to go through all the details of this because that will take up all the time, but in essence, what they basically show is that any architecture from 1 to 5 that senses an intermediate, what that does, in essence, is it upregulates the expression of enzymes downstream of the metabolite that's sensed. So that means that any reaction that's happening after the metabolite that is being sensed is upregulated by that metabolite. So if there's lots of it, it's saying that use me up as fast as you can, right? And the reactions that happen upstream of that metabolite, it's saying that you don't need to be upregulated as much, but the concentration needs to be on not producing me, but on consuming me to the end product. And that's the design principle behind those architectures. Now, the sixth architecture, where it senses the metabolite at the end, the product, is basically saying that if I have enough product, that you don't need to produce the enzymes to really produce me. Yeah, so basically slow down. And those are the design principles behind the amino acid biosynthesis and how it's able to adjust and adapt according to the need. Now, without going too much into the details of this because of time constraints, there are actually 2 different time scales on which this regulation is happening. The black-gray line that you see on the top of each of these architectures and the yellow and red lines which are happening at the bottom, they're actually working on 2 different time scales. The one at the top, this is known as a metabolite allosteric feedback. This happens very, very quickly because it's able to sense the product concentration directly and say, okay, slow down the first reaction of this whole cascade, which slows down the whole cascade. But the one at the bottom, that is happening on a much slower time scale. So what's happening there, it's changing gene expression, and gene expression happens much more slowly. And so it's saying that if there's a drastic change in metabolism and there's a drastic change in the demand for the amino acids, then we change the shift in the expression of the enzymes to cope with the demand and allow greater flux or less flux. And so we don't produce enzymes unnecessarily to waste other cellular resources. So it's an efficient process in essence. Okay, so I was just talking about metabolism and the regulation of metabolism. Now, even in metabolism, in metabolic reactions, there are regulations happening there, as I just explained. They happen on fast timescales. But what can happen is there can be a fluctuation of the input to these systems. Now, if we just concentrate on the two diagrams above the plots, the one on the left shows what's known as a feed-forward activation. So this is a metabolic reaction. There's an allostatic regulation going on here. And some metabolite X, biochemical X, actually feeds forward information onto a reaction downstream and says, become faster, like activate it, yeah, and use up X. Well, actually use up Z. The one on the architecture on the right, this is a feedback inhibition. So what this is saying is that if I have a product Z, it's saying if there's lots of it, then inhibit the upstream regulation and slow down the production of Z. Now, the work of Reed et al., and actually Ian Stewart, who I had the pleasure of meeting, they looked into what these regulations mean. And what these regulations are in essence doing, they're causing homeostasis. Homeostasis basically means the plots that you see on the bottom are basically giving the same message. They're saying that for an input I, which is on both of the x-axes, and an output Z, which is on the y-axis, if you have a change in the input at some intermediate range, Z will not change that much. Yeah? And for a feed-forward activation, there's a narrow range for which Z will not change for change in I. And for the feedback inhibition, there's a larger range for which Z does not change that much for change in I. And these are known as homeostasis. This is basically preserving or conserving the production of Z. So this acts as a way to filter out noise in the input. It's yet another design principle. Now, I realize that I ran out of time, so I'm just going to quickly jump over this. Noisy gene expression, I mentioned that earlier, because genes are expressed at very low levels, and so they can burst. And the cell has to cope with noise, but noise is not necessarily a bad thing. It can be very good. It actually is exploited in the cell to create heterogeneity in the population. What that means is that if cells behave in different ways, so if some cells grow very quickly and some cells grow very slowly, this is heterogeneity in the population. Now, heterogeneity is very good for survival, because if there is a very drastic change in the environment, some of the cells will not be able to cope with that. And so you will have other subpopulations of those cells which are almost pre-programmed to be able to cope in this new environment. But environment can change in many different ways, so there can be many, many different subpopulations. And noise is a way to actually help in producing phenotypic heterogeneity, like heterogeneity in the behavior of the cells. Not only phenotypic heterogeneity, but a recent study by Uphoff et al., Paulson's group in Harvard, they also showed that that certain regulatory actions inside the cell, which regulate the demethylation and damaging of DNA, that regulation actually also regulates the genotypic heterogeneity. So cells can actually become genotypically diversified, to help in their evolutionary adaptation to long-term changes in the environment. Now, the last slide, I just wanted to just jump over some applications of these learnings. So these learnings have really inspired my research. I'm using mathematical models to try and understand these regulatory mechanisms and their design principles, just as all of these authors have done, scientists have done as well. But now people are moving into the phase of applying them. They're using techniques in synthetic biology to try and re-engineer cells to harness them for our good. One of these is in biotechnology, where they're really harnessing these types of mechanisms, these transcription factors, which I call sensor effector in this figure. They're harnessing this to help redistribute metabolism to make the cell like a microfactory to produce a product of interest as opposed to growing. So they grow the cells to a large population, and then they say, well, you don't need to grow anymore, produce the product for me instead. And people are actually doing this, and they have been doing this in metabolic engineering for quite some time now. On the health and medicine side, we can use these regulatory understandings of these design principles and regulation to understand the dysregulation and the emergence of disease, like in atopic dermatitis. We can use them, they're now being used as diagnostic tools where they engineer bacteria, like E. coli, to excrete a certain chemical, which is just a coloring. It doesn't interact with our metabolism. It's just a coloring. They excrete that according to what chemical makeup is in our microbiome. And that can be an indication of certain diseases. So you could have, salmonella poisoning, or you could even have a stomach ulcer. And by consuming these bacteria, they will sense the environment and produce the color of interest, and then you can diagnose people like that. Another was cancer therapeutics. This was a really cool talk that I saw from the group of Jeff Hastley in Caltech. And he was doing some amazing work on using cancer therapeutics. So that's it. That's some of a summary of the design principles that I saw. And if anybody has any questions, please feel free. Jazakallah.
Jazakallah, Ahmad. I didn't understand anything that you used to talk about at Imperial 15 years ago, and unfortunately that's still the case today. But I saw lots of men down here scribbling in their notepad, so if you've got really quick questions and really quick answers, we can get through a couple of them. And of course, if any ladies have got any questions, please interrupt.
Just a quick question. You mentioned in slide 5 about external noise and environment and how that affects the cells. So is it also related to, I know it might be a different topic, but stress, does that have the ability to alter the gene as well? Like with external noise factors?
Well, there is this mechanism that I mentioned earlier, which is the regulation of methylation on DNA. So actually, if the cells, not actually under stress, but if the cells are growing very, very quickly, this protein is produced at very low levels, and so it can be diluted away. So there may not be any of these proteins there. And if there's any proteins, that means it doesn't demethylate the DNA, which can cause damage and cause genotypic heterogeneity. So it can allow the freedom of diversification.
No follow-ups. Any questions from the ladies? Okay, got one more from the men's.
Yeah, thank you for that really cool talk. I have a question about one of the things you mentioned. You mentioned when the DNA damage programs are activated, those actually promote heterogeneity among the cellular population. I can understand why it would be useful at a prokaryotic and even at a eukaryotic level, but it obviously creates a problem in the context of cancer. My question is, how does it actually manage to create heterogeneity when the same pathway is activated? One would think that you would activate the same things in every single cell, but how is it actually? Is it a probabilistic mechanism, or what's happening?
What do you mean? What is activated?
So you said the DNA damage pathways are activated.
Mm-hmm.
So presumably, if one pathway is activated, it would lead to the same endpoints. But you mentioned it leads to heterogeneity rather than the same endpoints.
Yeah.
So
No.
how does it actually happen?
Well, the heterogeneity it comes about over a very long time scale. It is not over a short time scale. So through many cell divisions, you could get genetic mutations, and this heterogeneity emerges over very long time scales,
So
not short time scales. It is not on a short time scale.
Okay.
No, it is active, but not on a short time scale.